Welcome to Project OpenASO. My name is Lela Lackey, and I'm the team lead for the OpenASO project. The purpose of this project is to use machine learning to develop an algorithm that can identify regions within an RNA that are suitable for targeting with an antisense oligonucleotide. ASOs are a useful research tool for scientists and can be developed into therapeutics. The idea of ASOs is very simple. You select an RNA you're interested in and create a complementary sequence to a region of that RNA. Then the ASO simply base pairs with the target. This makes nearly all RNAs potential targets in a specific manner. However, selecting the region to target with an ASO can be tricky. For example, RNA binding proteins or microRNAs may already be bound to an RNA and block the ASO from binding. Highly variable positions, ones where there are common single nucleotide variants in the DNA, perpetuate into RNA and can decrease the ability of an ASO to bind. Likewise, after transcription, RNA bases can be modified. One of the most common modifications, like A to I editing, where adenosine nucleotides are converted to inosine, makes the nucleotide behave more like guanosine and alters how it would interact with the ASO. In addition, the end of a messenger RNA is determined by polyadenylation sites. Many RNAs are shorter than their documented length due to alternative polyadenylation. Designing an ASO that targets the end of a messenger RNA has to take this into account. Finally, RNA is not a linear molecule. It forms structures that influence the ability of an ASO to interact with the target region. Identifying accessible regions is essential for picking an effective ASO target. Nucleotides that are already base paired are not available to interact with an ASO. The overarching goal of this project is to use machine learning to identify regions suitable for ASO targeting within an RNA. One specific question I'm interested in is how RNA structures should be incorporated into ASO selection to capture useful information about accessible regions. The question is whether complex ensemble models of RNA structure perform better than simple models, such as minimum free energy models, for target selection. What I mean by that is, often when researchers are designing ASOs, they look at a single RNA structure, usually the minimum free energy model, the most energetically favorable, and they use that to identify accessible regions. However, RNA is a very flexible molecule and is better represented as an ensemble of structures. So while the most favorable structure, in the middle here, may exist in the cell, other slightly less favorable structures that shift the accessible region may also exist and influence ASO binding. To develop a predictive algorithm for ASO design, we will use a dataset of known ASOs and their effectiveness, and combine that with features that we know are important for ASO targeting, like RNA binding protein sites and RNA structure. Currently, we have an ASO dataset containing around 3,400 ASOs for 100 genes. This is a histogram showing the number of ASOs targeting each gene. Most genes have around 60 ASOs. If we look at a graph of the effectiveness of the ASOs against each gene they're targeting, we can see that the effectiveness of ASOs varies from around 30% to nearly 100% suppression ability on average between different genes. However, for each gene, there's a spread of effectiveness, indicating that some ASOs are better than others. If you're interested in this project and would like to contribute, I would love to have you join our team. Even if you only have minimal bioinformatics experience, you can help this project by identifying key data sets and helping process them into a suitable form for machine learning. Anyone with experience in RNA secondary structure modeling would be very helpful for answering the specific question of how RNA structure information should be best incorporated into ASO target selection. Individuals with machine learning experience are key for completing this project and linking the features we're interested in with ASO effectiveness. Finally, if you have experience creating graphics to display scientific results, your experience would be welcome to help visualize our work. Individuals with the ability to create accessible interfaces to explore the data and the algorithm would also be welcome. If you're interested and you want to learn more, check out the sample data on GitHub. There are also plenty of databases to explore. Feel free to check out the listed datasets here or find new datasets of your own. In addition, for further reading, there are 2 specific papers related to this project listed here. I'd love to have you as part of the team, so if you think this project sounds interesting, join us. Email me at lackeylela@gmail.com or contact the HackSeq 2020 team. I look forward to working with you.